Ashanath, wait.
Madam, online already started, madam.
Okay.
We start
So
the
let me
class.
start. Okay. Okay. Good morning, students. Myself, Kirti, I am working as JL in Tsrjc Sarvail. Here we have discussed about the human reproductive system, the male female reproductive system, the spermatogenesis and the gametogenesis, oogenesis. Then we have discussed the menstrual cycle, and we have also discussed the fertilization process of implantation, then bilaminar embryonic disc, process of gastrulation, then trilaminar embryo. Then we have discussed the formation of notochord, neurulation, that is formation of nerve cord or neural plate. Then differentiation of mesoderm. Here we were discussing yesterday where the class ended. So I'll just give a brief intro about this again. So the differentiation of mesoderm. The mesoderm, what structures it is going to give is given here. The intraembryonic mesoderm spreads all the direction between the outer ectoderm and the inner endoderm. So here we can see the the part which is present in between the ectoderm and endoderm. This is the endoderm and this part is the ectoderm. So the region which is present in between, that is this region, is known as, these regions is known as mesoderm. Now, the mesoderm derives to give certain structures in our body. So here, the mesoderm at the embryonic stage, that is at the early embryonic stage, is classified into epimer, mesomer, and hypomer, which we have learned in the previous sessions. So these epimer, hypomer, and mesomer, their location is, the location of the epimer is near the notochord, on the sides of the notochord, that is on the longitudinal axis of the notochord, whereas the location of the mesomer is around the, okay, the location of the hypomer is around this alimentary canal, which is this archenteron, which is going to form the future alimentary canal. And in between this hypomer and epimer, there is a structure which is known as mesomer. So these structures are the earliest differentiation of mesoderm tissue. Later on, these epimer, mesomer, and hypomer are only going to give rise to certain structures of the body. Here we can see the epimer becomes segmented into cuboidal blocks called somites or metameres, and each somite differentiates into sclerotome, myotome, and dermatome. These sclerotome forms the vertebral column, we know, which is the backbone of the body and which helps in there are various functions, which is a form of it is also a form of exoskeleton, endoskeleton in a body. Then myotome, which forms the voluntary muscles. So voluntary muscles are nothing but the skeletal muscles of the body, which are attached to the bones, and they help in providing movement to the body. And voluntary muscles are under our control. That's why we call them as voluntary muscles. The third one, that is dermatome. The name itself says derma. Derma is nothing but related to skin or dermis. So the dermatome forms the dermis of the skin. That is, we know that the skin is made up of many layers. The upper layer is epidermis, and below the epidermis there is a layer which is known as dermis. Now this epidermis is derived from the ectoderm, whereas dermis is derived from the mesoderm, because this ectoderm, the ectoderm. Okay, let's talk about the dermis only. So dermis is the one which is having a rich supply of blood vessels as well as the lymph vessels, and there is a supply of nervous tissue also to the dermis layer, whereas there is no supply of blood vessels or nervous tissue to the epidermis. The mesoderm forms the urinogenital organ and their ducts. So urinogenital organs are also formed from the mesoderm. Next, the hypomere splits into outer somatic and inner splanchnic mesoderm, which are nothing but the lining of this outer somatic is the lining of body cavity, and splanchnic mesoderm is the line, or also known as visceral mesodermal layers. These are the lining of the visceral organs which are present in the body. Intraembryonic coelom is formed between these two layers. So the coelom is present in between somatic, outer somatic and inner splanchnic mesoderm. And in the coelom only, it gives space for pericardial cavity, pleural cavity, and peritoneal cavities. Now you can see here in this diagram, all the structures are very clear. Epimere, which is giving rise to dermatomes, sclerotome, and myotome, whereas hypomere giving rise to somatic hypomere and splanchnic hypomere. And then in between this somatic and splanchnic hypomere, you can find the coelom. And in the coelom only, all the visceral organs are displaced in our body. Then you can see here epiderm. Epiderm is going to be which is derived from the ectoderm, and this part is the archenteron, and this is the endodermal endoderm with the yolk present here. But in humans, the amount of yolk is almost negligible, so that's why the condition is known as microlecithal condition. And then mesomere is the one which is going to give rise to the mesoderm. Next, let us see the extra. So this is about all the structures which are going to be formed. Now all these structures are surrounded by extra embryonic membrane. So this is complete embryo. This structure starting from the epiderm to the endoderm, and the things present inside, all the organs or all the tissues whichever are present inside. This is the embryo of the organism. Now this embryo is covered by extra embryonic membranes, and these extra embryonic membranes have certain functions. The extra embryonic membranes helps in protection of embryo from desiccation, mechanical shock, absorption of, and it also helps in providing absorption of nutrients and exchange of gases. So these are the main four functions of the extra embryonic membranes. Now, what are those extra embryonic membranes? They are chorion, amnion, allantoic, and yolk. Four extra embryonic membranes surround from desiccation, mechanical shock, and it also allows absorption of nutrients and exchange gases, exchange cycle. The in the earlier classes we have learned where the epiblast is in the bilaminar embryonic disc. We have seen the outer layer was epiblast, inner one was hypoblast. This hypoblast was present around the which yolk appears. Adjacent to the trophoblast, the outermost layer, might system migrates away from trophoblast, forming the amniotic cavity. Now, this is amniotic cavity present in the amniotic, which is present in the amniotic, which is made up from the blast. Now, if this is the amniotic cavity from blast cells. These aminoblast cells are amniotic cavity, which is okay. These aminoblast cells are derived from the epiblast cells. We have seen bilaminar embryonic disc. This amnion is filled with cavity. Extra embryonic membranes in the next these
ఈ స్టోర్ రూమ్ చేస్తే ఎలా ఉంటుంది?
extra embryonic membranes embryonic membranes here embryon and yolk sac. These are embryonic membranes. You see, the main function of protects the
స్టోర్ రూమ్ ఇప్పుడు స్టోర్ రూమ్ కి ఎన్ని ఐటమ్స్ సరిపోతాయి? పైదాకా అట్లాంటి గ్రిల్ పెట్టించారు, లేకపోతే
రాజ్కుమార్ సార్, రాజ్కుమార్ సార్,
అదే నేనని
రాజ్కుమార్, రాజ్కుమార్ సార్,
ఇలానే పిల్లలు కార్డ్స్ చేస్తే
అది కూడా
రాజ్కుమార్ సార్.
Ma'am, can you mute everyone? I mean, others. 1 second, let me.
కొంచెం విగినా కూడా వాళ్ళకి
వచ్చి కూడా అట్లాంటి ఫిల్స్ దొరుకుతాయి మేడం. కాస్ట్ 1000 అంటే మరీ తక్కువ. సరే అది నిజమే.
రాజ్కుమార్ సార్, రాజ్కుమార్ సార్,
సారీ మేడం చెప్పండి.
మ్యూట్ మ్యూట్ ప్లీజ్ మ్యూట్ ఇట్. Yes, Kirti, please continue.
Yes, so we were discussing about the extra embryonic membranes. Now, these extra embryonic membranes are nothing but amnion, chorion, allantois, and yolk sac. And the main function of these membranes are amnion membrane is mostly used for protection, and allantois is for exchange of material as well as exchange of sections, that is exchange of carbon dioxide and oxygen. Then the third layer is chorion, which is also helpful for gases exchange, and the fourth one is yolk sac. Now, from which layers they are derived, we will see. We have seen that in the earlier one, we have seen that amnion is derived from the epiblast cells, which is formed from the amnioblast cells. So epiblast cells will be giving rise to amnioblast cells, and these amnioblast cells will be forming the amnion layer as well as amniotic cavity. So the amnion layer is derived from the epiblast cell. The next structure, the hypoblast is pushed down, forming the lining of cavity called yolk. So in the earlier class also we have seen this is the yolk sac, this is the yolk part, and in the bilaminar disc we have seen that the hypoblast cells are moving down by the process of ingression, and they are lining the cavity of the yolk sac. So the cavity of the yolk sac is formed by the hypoblast cells. Next is chorion. The chorion consists of outer layer formed by primitive ectoderm or trophoblast. So the chorion layer is derived from the primitive ectoderm or the trophoblast cells, which are the outermost cells of the embryo, which were formed during the starting stage. We have seen that in the starting stage, the embryo was formed with trophoblast cells, which were forming trophoblastic villi, as well as inner mass cells. Now, these inner mass cells are the ones which are going to give rise to the epiblast and hypoblast, and later on epimere, hypomere, and mesomere. And these trophoblast cells, which are the outer cells, they were meant to be forming the extra embryonic membranes. So the chorion is formed from that primitive ectoderm only, that is the trophoblast cells, and the inner by somatic mesoderm, from which chorion cavity is formed. So this chorion layer is derived from two types of germinal tissue. One is primitive ectoderm or the trophoblast, and the cavity of chorion, the cavity which is present around the chorion, is derived from the somatic mesoderm cells. We have seen somatic mesoderm cells, it is derived from the hypomere, right? The hypomere is forming the hypomere was forming the somatic mesoderm as well as splanchnic mesoderm or visceral mesoderm. So that somatic mesoderm is also giving rise to the cavity of the chorion. So we have to remember all these terms, which cell is derived from which one. So the chorion undergoes rapid proliferation, that is, it undergoes mitotic division and forms chorionic villi. Now this chorion, if this is the chorion layer, then it undergoes division, mitosis division, and it will be forming the chorionic villi-like structure, which will be helpful in absorption of nutrients or the exchange of gases by the process of diffusion. So here, as we know, the function of the chorion is nothing but exchange of gases. The chorion villi invades the endometrium, that is the wall of the uterus, which is a part of mother uterus, which is a part of the maternal body. So this chorionic villi invade the endometrium and help in transfer of nutrients and oxygen from maternal blood to the fetal blood. So that is the function of chorion. It helps in transfer of nutrients and oxygen from the maternal blood to the fetal blood. So next slide. Okay. Now here, let us see the next part. Chorioallantois. Now the chorion, see here, you can see the chorion like or the wall of the endometrium to absorb the nutrients as well as the exchange of, and this is the amniotic cavity. Okay, first let us discuss the function of this chorion. The chorion surrounds the embryo and other membranes. Soon a fourth membrane called allantois develops as an outgrowth from the embryonic hindgut. Now embryonic hindgut is nothing but the it is the one which is derived from the archenteron layer. So it means that it is derived from the endoderm. So the membrane of the allantois meets the inner surface of the chorion and forms a highly vascularized region called allantochorion. So the chorion as well as the allantois both will together form a highly vascularized means which is having a rich supply of blood vessels. So this will be forming a highly vascularized region which is known as allantochorion. Now this allantochorion contributes the fetal part of the placenta. Now we know that there is placenta which is helpful in exchange of gases and transporting substances from mother to the fetus, and transport of excretory waste from fetus to the mother. So that placenta is made up of maternal placenta as well as fetal placenta. So this allantochorion is the one which is contributing the fetal part of the placenta. The placenta facilitates a more effective and efficient part of exchange of nutrients, respiratory gases, hormones, antibodies between fetus and mother by 12th week of pregnancy. So once the placenta is formed, then the placenta will be performing all these functions, as well as it will also take over the function of corpus luteum. We have seen that corpus luteum will be remaining. If there is fertilization, the corpus luteum persists for 3 months. Now that corpus luteum, if there is no pregnancy, will be converted into corpus albicans, which doesn't release any hormone, whereas corpus luteum will be releasing the progesterone hormone. It will be there only for 3 months, which is known as corpus luteum of pregnancy. Now that corpus luteum will be regenerated after 3 months, and the function of corpus luteum, that is releasing of progesterone hormone, will be taken over by this placenta. So after that, after the 12 weeks, that is 3 months of pregnancy, this placenta is completely formed, and it takes over the function of corpus luteum, as well as it also facilitates the efficient exchange of nutrients, respiratory gases, hormones, antibodies between fetus and mother. Now here you can see the clear structure of all the things which are present in the embryo. The yolk sac, you can see this is embryo. This is the embryo, and you can see here the umbilical cord, and this embryo is placed in the amniotic cavity, which is released from the amnioblast cells of the epiblast, and this amniotic cavity will protect the embryo from desiccation or from the mechanical shock. Then this yolk sac, this is the yolk sac, and these are the chorionic villi, which will be helpful in transporting the substances or exchange of gases. And this is the uterus part. So these are the extra embryonic coelom. These are the embryonic coelom which is present outside or in between the extra embryonic layers. Now the next thing is placenta. So placenta is a tissue which is formed. Placenta is a tissue which is formed in between the mother and fetus for various functions. It is the connection between the mother and the fetus. Now, placenta is made of 2 parts. One is maternal placenta and the other one is fetal placenta. Maternal placenta is derived from the endometrium of the uterus, and fetal placenta is derived from the fetal part of the fetus, that is from the developing embryo. The maternal components of the placenta are uterine epithelium, uterine connective tissue, and uterine capillaries. So epithelium is the one that is outermost layer of the uterus, then uterine connective tissue, and the uterine capillaries, that is artery as well as veins. So these are the ones which will be forming the maternal component of the placenta, that is from the mother. And the fetal part of the placenta is, the components of placenta are fetal capillaries, endothelium, fetal connective tissue, and fetal chorionic epithelium. So chorionic epithelium we have seen, which is helpful in the one which is producing the chorionic villi, and this is the one which is helpful in forming the fetal part of the placenta, right? So the fetal part of the placenta is formed from these tissues: fetal capillaries, fetal endothelium, fetal connective tissue, and fetal chorionic epithelium. The Chorioallantoic placenta, the inner region of chorionic villi, develops a network of capillaries of umbilical artery and umbilical vein. So these umbilical artery and umbilical vein will be developing a network of capillaries, and these vessels run through the umbilical cord. The placenta of human is called chorioallantoic placenta, as allantois also fuses with the chorion in the process of vascularization. So chorion and allantois both are fused to form the placenta here in humans, whereas it is a different case in other organisms. So that's why we call them as chorioallantoic placenta. And they will be helpful in the process of vascularization, that is supply of blood vessels. Placenta is described as hemochorial placenta, as the maternal blood comes into direct contact with the membrane of fetal chorionic villi. So that's why it is known as hemochorial, because the blood vessels of the mother, or the blood of the mother, is coming in direct contact with this chorionic villi. If these are the chorionic villi, then the blood is directly coming in contact with this. That is why it is known as hemochorial placenta in humans. That pregnancy will start as soon as the is embedded in the uterus. So it will be also called gestation period, which human pregnancy about 26 days from Hello. Hello.
Hello. Kirti,
Ma'am,
what
give me
happened?
host button.
Oh. Kashinath? 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 Kashinath?
Ma'am, give me host button. Share again.
Kirti?
మేడం ఆన్లైన్ లో లేరు మేడం.
Just now she dropped.
అవును మేడం.
Wait.
ఓకే మేడం.
Do me host.
ఓకే.
The placenta.
మేడం షేర్ యువర్ స్క్రీన్ మేడం. మేడం వాయిస్ రావట్లేదు, మళ్ళీ వెళ్ళిపోయారు మేడం.
Kirti?
మేడం మేడం వాయిస్ రావట్లేదు మేడం. కీర్తి మేడం మీ వాయిస్ రావట్లేదు, మైక్ లేదు మేడం.
Hello.
మేడం షేర్ యువర్ స్క్రీన్ మేడం. అరే
Hello.
మేడం షేర్ యువర్ స్క్రీన్ మేడం.
There is some network problem, sir. Here, it's
Okay. No issue madam. Continue madam.
coming. Coming, sir? Is it visible?
Yes.
Yes
The
madam.
screen is
Yes.
coming.
Yes.
Continue madam.
Okay. Okay. So the functions of placenta. It supplies oxygen to the embryo and removal of carbon dioxide and excretory waste produced by the embryo. So, the main functions of the placenta. The placenta is connected to the embryo through an umbilical cord, which helps in the transportation of substances to and from the embryo. Next, the provided by the placenta and is essential for the maintenance of pregnancy after the fourth month. Already we have discussed. That is, it is taking over the function of corpus luteum. The function of the corpus luteum is to release the progesterone. It reaches the maternal blood and promotes uterine growth of the rest of the functions of placenta is human chorionic gonadotrophin, that is HCG, used by the placenta.
Please share
And
your
the function of this HCG, my screen is not coming, ma'am. I have
Yes,
shared
now-
already.
Okay, now it is okay. Carry on.
Okay. Okay. So human chorionic gonadotropin, that is HCG, is produced by the placenta, and the functions of this is similar to that of the luteinizing hormone. And human chorionic gonadotropin sends the message of pregnancy to the master endocrine gland, that is the pituitary gland, so that LH is secreted to maintain the corpus luteum and prevents menstruation and further ovulation. So this HCG is released from the placenta, and it sends the message to the it sends the message to the pituitary gland. This pituitary gland will in turn release the LH hormone, which will be helpful in maintaining the corpus luteum for the first 3 months, and which releases the progesterone hormone, which is essential for the formation of, or which is essential for the maintenance of the pregnancy. The gonadotropins are excreted. What are the gonadotropins? Already we have discussed, that is FSH and LH. So FSH and LH are the two gonadotropins which are excreted through the maternal urine, where their presence in the urine is used as a test to detect the pregnancy in the earlier stages. So due to presence of these hormones, that is human chorionic gonadotropin as well as the FSH and LH, some amount of these hormones will be present in the maternal urine when there is pregnancy. So these are the indicators of pregnancy at the very earlier stages. So when they go for a pregnancy test, these are the things they are going to check for. That is, in the urine sample, they will be finding out what is the level of FSH and LH, by which the earliest stages of pregnancy can be detected. Next, somatotropin. Somatomammotropin is also called placental lactogen, and it has an anti-insulin effect. Means it is acting against the insulin hormone in the mother and leading to increased plasma level of the glucose. That is, generally the function of insulin is to convert the glucose into glycogen and to store it in the liver. Here, what this somatomammotropin is doing, it will act against the insulin. So it is an anti-insulin hormone which acts against the insulin, and it will reconvert, or it will not allow the insulin to convert the glucose level into glycogen, because the mother body, because there should be more amount of glucose in the mother's blood so that it can be available for the developing embryo. So it increases the plasma level of glucose and amino acids in the mother circulation, so that these amino acids as well as glucose can be transported through placenta to the developing embryo. In this way, it increases the availability of these material to the fetus. So these are the main functions of the placenta. The most important thing is human chorionic gonadotropic hormone, which we have to remember. Next, the placenta protects the fetus from the relatively high blood pressure of the maternal circulation. The fetal tissues having a Paterbach chromosome effect acts as foreign structure to the mother, who produces antibodies. And the fetus sustains such a rejection of about 38 to 40 weeks by producing certain immunosuppressant substances. The fetal tissue is having some chromosome effect, this Paterbach chromosome effect, and it is acting as a foreign substance, which the mother, by which it is acting as a foreign substance to which the mother can produce antibodies. Now, if these antibodies are produced, and if these antibodies are going to the fetal body, then it may result in damage of the cells of the fetus. But the fetus will sustain such rejection for the gestation period by producing immunosuppressant substances. So these immunosuppressant substances are acting as a protection barrier for the fetus against the antibodies which are produced from the female, or which are produced from the maternal body. The next is, let us see the timetable of events during the pregnancy. So as we know, human gestation is divided into 3 trimesters of about 3 months each. So it's for 9 months. So each 9 month is divided into 3 trisemesters, and each trisemester will be having 3 months. The first, let us see the developmental stages of these events which occur during every trimester. The first trimester is the main period of organogenesis. That is, organogenesis is nothing but formation of the organs. So in the first trimester, that is first 3 months, organs will be starting to form or starting to generate. And then heart is formed if just after the first month, that is completion of the first month or 1 month of pregnancy. The heart will be very well developed, and it can be checked in the where it can be checked by stethoscope. By the end of the second month of pregnancy, the fetus develops limbs and digits. So forelimbs as well as hind limbs with their digits are also well developed by the second month of pregnancy. By the end of 12 weeks, that is 3 months, most of the major organ system, that is digestive system or urinogenital system or all the type of systems are very well developed by the 12 weeks, that is 3 months of pregnancy. And the genitalia are also very well developed. So female genitalia or male genitalia, depending on the sex of the individual, are also very well developed by the end of 3 months of pregnancy. So we can say the first trimester, almost all the organs are very well developed. The first movement of the fetus and the appearance of hair on the head are usually observed during the 5th month. So it can vary as well. That is, first movement when the fetus starts to move inside the mother's embryo will be observed during the 5th month, and the hairs or the development of hair follicles on the body is also started in the 5th month. Next is stages of development. So here you can see by the end of 24 weeks, the body is covered with fine hair, eyelid separates. The eyelids are already formed, and now the eyelids are separated. So the embryo can see, and eyelashes are also formed. By the end of 9 months of pregnancy, the fetus is fully developed and is ready for delivery. Now you can see here at various developmental stages: 3 weeks, 4 weeks, 5 weeks, 6 weeks, then 9 to 12 weeks. Almost all the organs are formed here, and the fetus is starting to grow. So all the organs are developing. All the organs are already developed by the 12 weeks. Now the fetus will be growing into a mature fetus. After the complete formation of the fetus, the next step is parturition. So the process of delivery of fetus is called parturition. The parturition is induced by a complex neuroendocrine mechanism. So neuro as well as endocrine mechanism, that is brain as well as endocrine glands, they are together coordinating with each other to induce the process of parturition. The signal of parturition originates from fully developed fetus and placenta, which induces mild uterine contractions. As we know, the uterus is made up of muscles. Smooth muscles are there. That is, myometrium of the uterus is made up of smooth muscles, and these smooth muscles will start contraction. So mild contraction of the uterus will be started, which is known as fetal ejection reflex. So due to this contraction of this fetal ejection reflex, this triggers release of oxytocin hormone from the maternal pituitary. That is, from the pituitary gland, oxytocin hormone will be released by this contraction of the uterus, which gives fetal ejection reflex. Then oxytocin acts as uterine muscle and acts on uterine muscle, and it causes strong uterine contraction. So again, more and more uterine contractions will be started, which again in turn stimulates the release of oxytocin. So oxytocin will be released and results in the uterine contraction. This uterine contraction again will induce the pituitary gland to release more oxytocin. So it is a continuous process till the birth. This leads to expulsion of baby out of the uterus through the birth canal. So once the baby is completely developed because of this uterine contractions as well as oxytocin hormone, this process of parturition will take place. And soon after the birth, the placenta along with the decidua is also expelled out of the uterus. Here you can see the process of parturition, that is action of giving birth or the childbirth. And in the normal vaginal delivery, the fetus will be born. And if the fetus is unable to come out by its natural process, then there will be cesarean. So after the delivery, the next step is lactation. Now, lactation, we have already learnt about the mammary glands, which is the one which will be releasing the milk or the colostrum, which is the first form milk. One aspect of postnatal care, that is after the birth, unique to mammals is lactation. So because of the presence of the mammary glands only, the mammals are known as mammals. The name mammals is given because of the presence of mammary glands, and with which they show the parental care uh, to the individual. So aspect of care which is unique to mammals is lactation, that is production of mother's milk, which is produced from the mammary gland. Hypothalamus signals the anterior pituitary hormone milk. So the hypothalamus is acting on the gland, and this protein hormone will be stimulating the mammary to release the, and the processes will be occurring only after the process of partition. Mammary glands will undergo differentiation pregnancy and starts producing milk towards the end of the pregnancy. Then we have already discussed about this mammary glands, how it is, what are the structures of mammary glands, and how it produces the milk. This helps the mother in feeding the newborn. The milk produced during the initial few days of the lactation is called colostrum, and this is the first formed milk, and we know it is very much nutritious because it contains very essential nutrients, as well as there are several antibodies, and the type of antibodies which is seen in the colostrum is IgA, which is essential for protection of the newborn against the diseases. So that these IgA type of antibodies are the ones which are helping in the embryo to protect it from the infectious
Madam, conclude the session, madam.
or the one second is very much essential for the embryo. So that is about the lactation, and with this we have the human reproduction chapter. So tomorrow I'll be starting the next chapter.
Thank you,
Okay,
madam.
sir.